everyone. Welcome to our February 9th EMSL LEARN webinar, where today we'll be discussing the currently open EMSL JGI FICUS call for proposals, which includes access to capabilities at the Center for Structural Molecular Biology at Oak Ridge National Laboratory and the National Science Foundation's National Ecological Observatory Network, which is also known as NEON. We are so glad to have you join us today for this EMSL LEARN webinar on the resources that are available through the open EMSL JGI FICUS call for proposal. Our next EMSL LEARN webinar is on February 23rd, and it's going to be focused on the capabilities at EMSL to analyze mineral and organic surfaces. So we hope to see you there. Also, make sure to keep in touch with EMSL by subscribing to our email and social media channels if you haven't already. So the links will be in the chat. It's a great way to keep up to date on our open call for proposals and any other opportunities that we have available for our users. Today, we begin with EMSL chemist and our integrated research platform leader for biomolecular pathways, Mary Lipton, who will give an overview of the capabilities EMSL offers for this FICUS call. Mary will also conclude today's presentations with an overview of tips for submitting a successful proposal. After Mary, Tanja Woyke, the Deputy Director of JGI's user programs, will give an overview of the capabilities JGI offers for this FICUS call. Hugh O'Neill, a senior scientist and group leader at the Neutron Scattering Division at Oak Ridge National Laboratory, will provide an overview of the biological small angle scattering capability at the Center for Structural and Molecular Biology offered through this FICUS call. Mike San-Clemens, the lead research scientist at NEON, will give an overview of the NEON biorepository users have access to through this FICUS call. Trent Northern, a senior scientist at JGI and Deputy Director of Environmental Genomics and System Science Biology at Lawrence Berkeley National Lab, as well as Kirsten Hofmackel, Earth Scientist and Microbiome Science Leader at PNNL, we'll be discussing the fabricated ecosystems capabilities available at EMSL and JGI and how you can participate in a ring trial through this FICUS call. If you're interested in submitting a proposal for this open FICUS call, please be aware of the following dates. You'll need to submit a letter of intent by March 16th. The letter of intent provides FICUS reviewers with information about your proposed project details, participants, funding, resources, and the samples needed. The link to our webpage with more information about the letter of intent can be found in the chat window. If your letter of intent satisfactorily shows that your project is responsive to the call's focus topics and it's aligned to BER science mission, then you'll be invited to submit a full proposal. Full proposals are due on May 4th. There will be time for questions after these presentations, so we encourage you to post your questions in the Zoom chat window. Our speakers will then answer them during the moderated Q&A session. And so with that, we will begin with Mary.
Thank you, Linda, for the introduction. Today, what I want to talk about is the available EMSL resources for FICUS projects. And just put it into a little bit of context as far as the way that EMSL is organized. So EMSL has organized into what I have here is two science areas, and we have a third one that's kind of represented at the bottom. The first one is focused a lot on biology, and the second one on environment, and the third one is our new computation and modeling IRP, or science area. The science areas are then further broken down into what we call integrated research platforms. And the integrated research platforms are really focused on the science that we want to do within each of these science areas. And so The biology and environmental science areas are both broken down into 3 different IRPs that I'm going to go through briefly. And right now we have one integrated research platform for the computation and modeling. So a closer look at the biology science area, like I said, we have 3 integrated research platforms. The first one is biomolecular pathways, which I am the lead for, and we're basically focused on using advanced analytics and omics capabilities to discover the principles underlying the translation of genomic information into metabolic pathways, regulatory networks, and cellular function. Our structural biology integrated research platform, led by Scott Lee, generates structural, biochemical, and dynamic information about proteins, protein complexes, other biomolecules, as well as looks at cells on a holistic level. Our cell signaling and communications integrated research platform is led by Alex Belyaev. And what we want to do there is pursue an understanding how complex phenotypes are impacted by cell-cell communications and how those communications occur on a molecular level, but then also impact the entire system. Our environmental science area is also broken down into 3 integrated research platforms. Our plant and ecosystem phenotyping integrated research platform is led by Amira Kami. And what we want to do is aim to dissect the plant genotype environmental interactions. And this includes plant microbe interactions, rhizosphere phenomenon, as well as phylosphere phenomenon. And we have the ability to grow plants, look at intact phenotyping, as well as do stabilized tope labeling experiments in our plant growth chambers. Our biogeochemical transformation IRP is led by Emily Graham, and what we want to look at is a detailed molecular biogeochemistry within microdomains in soils and sediments. And she's actually right now leading a 1,000 Soils project, which is looking at these kind of biochemistries within soils across the country. Our last integrated research platform in the environmental science area is the ecosystem interfaces, led by Jim Moran. And what we have is a multidisciplinary expertise in analytics focused on looking at the transport of nutrients and materials that contribute to the biogeochemical processes. We're looking at the flow of nutrients through a system using stabilized tope labels, as well as just an overarching view of the atmosphere and how atmospheric interactions occur within our ecosystems. But I want to spend most of the time on our newest science area, and that's the computing analytics and modeling, or CAM. This is led by Jay Bardhan, who right now is both the science lead and the integrated research platform lead. So CAM is focused on using state-of-the-art experimental data to develop the predictive understanding of biological and environmental systems. We use advanced data analytics, visualization, and computation modeling and simulation. And it uses our newest supercomputer that was just brought up in this past year to bring a lot of the analytics together to integrate data as well as visualize data. And so some of the things we're looking at in this IRP is delivering computation and data analytics expertise for developing a predictive understanding of our biological and environmental systems. We want to do this on a multi-scale system, so we can get multi-scale models. We can look at processes across temporal and spatial scales. We want to maximize the methods to analyze and visualize our data. A lot of times users can get spreadsheets of data, but it's so much more impactful when we can see this either in a visual model or in some sort of conceptual design that helps us understand it a little bit better. And then if we can have this design go on the spatial and temporal scales, then it allows the human brain to actually see the data much better than just looking at spreadsheets. And then we want to deploy and develop software to analyze and visualize all of these extensive and complex data. So with that, please feel free to put any questions in the chat. I've put our contacts here, and I've noticed that Genoa, thank you, have put all of the contacts also in the chat. So if you're interested in any one of these, you can contact the people directly, but also feel free to reach out to me for any questions, and I can always point you to the correct people. And now I'd like to introduce our next speaker, who is Tanja Woyke from the JGI.
All right, thank you, Mary. And thanks to Linda for giving a very nice introduction. So in the next few minutes, I'd like to give you an overview on the resources that the Joint Genome Institute has available as a Department of Energy funded user facility through the FICUS call. Move forward here. Okay, so at a very high level, the JGI provides access to nucleic acid sequencing, both DNA and RNA, for which we use both next generation short read sequencing platforms, as well as third generation single molecule long read sequencing capabilities. We also offer DNA synthesis capabilities, and lastly, mass spectrometry based metabolomics, complementary to what EMSL offers. Sorry. As for sequencing, our core capabilities are focused on generating de novo genomes from plants, algae, fungi, bacteria, archaea, and viruses, which also includes their annotation. Comprehensive transcriptome analysis, which can be for either gene expression experiments or for improvement of annotations for our eukaryote genomes. Microbial community, DNA, RNA sequencing and annotation, so metagenomics and metatranscriptomics. And then lastly, resequencing for variant detection, which could, for example, be a laboratory evolved bacterial population for which we provide data on SNPs, single nucleotide polymorphisms, and small indels, insertion deletion events. We also have more custom capabilities available, and these include fluorescent activated cell sorting for targeted metagenomics and single cell genomics. For example, we can use BONCAT, which is bioorthogonal non-canonical amino acid tagging, which allows us to pull out translationally active cells from environmental samples and sequence these as so-called kind of mini metagenomes of the active community. We also offer stable isotope probing enabled metagenomics, and include the fractionation step as part of that being performed at the JGI. Whole genome DNA methylation analysis, specifically using the PacBio platform. And lastly, we offer DAP-seq, which is an approach that's been developed more recently at the JGI for the discovery of transcription factor binding sites. For our metabolomics capabilities, we offer LC-MS, MS-based metabolomic analysis of polar metabolites, for example, organic acids, amino acids, sugars, nucleobases, etc., and non-polar amino acids, for example, secondary metabolites and lipids. And for the DNA synthesis capabilities that we offer, these involve gene pathway DNA synthesis, and this can also include fairly large biosynthetic gene clusters, for example. We offer whole genome guide RNA library construction and QC, and organism engineering, and that is primarily through the CRATE system that was developed in-house. What I'm not listing here, but is also offered, are the Ecofabs, which are new, really exciting addition to the FICUS call, and you'll hear more about these shortly. Now, what is the maximum scale of the request that you can make? A PI can request up to 1 terabase of sequence data, up to 50 KB of synthesized DNA, and up to 200 or 500 samples for polar, respectively non-polar metabolomic analysis. You should select the amount that's best aligned with your scientific goals for your proposal, and we do encourage you to mix and match, exploit a combination of the different capabilities that we offer. And lastly, I want to show some point of contacts for you for specific questions. Please always come to Krista Panakia and myself first. Krista is the lead of the project management office at the JGI. She can answer any questions that relate to the logistics of your proposal submission. If there are in-depth scientific or technical questions, we will parse them out to the respective scientific program and technical leads that are shown here. Jeremy Schmutz, John Vogel are our plant program heads. Igor Grigoriev heads the fungal and algal programs. I'm responsible for microbial genomes, and Emily Ilo-Fadrash for metagenomes. Yasu Yushikini heads the synthesis science programs, and Trent Northern our metabolomics platform. I also have listed here 2 technology leads, which is Rex Malmstrom, who runs, for example, the single cell and the DNA zip platforms, and Ronan O'Malley, who's heading sequencing technologies, and who, for example, developed the DAP-seq approach that's available through the FICUS. And with that, please put any questions that you have in the chat, And I will hand it over to Hugh to tell you more about the CSMB questions, capabilities. There we go. You're muted, Hugh.
Oh, sorry. Okay, now I'm unmuted. I apologize. So my name is Hugh O'Neill. I'm from Oak Ridge National Lab, and I lead the Center for Structural Molecular Biology here. And I'm going to tell you today about how you can use small angle neutron scattering as part of your research capabilities. So if I can, yeah, there we go. So the SAMS provides, it's a structural characterization technique, and it provides structural information on the 1 to 500 nanometer length scales. So it can be used to study a wide variety of biological systems. So for instance, we can look at how proteins interact with their binding partners, such as other proteins or nucleic acids. It's very useful for studies of systems like biomembranes, where we can actually look at lipid assemblies in vitro, or even impact membranes in a living cell. And because neutrons are a non-destructive technique and highly penetrating, they can also be used to look at very complex systems. So you can look at soil structure, and for instance, how, if you were interested to see how nanoplastics interact in soil, or even just soil structure and its morphology. In addition, you could also look at plant cell walls. So using SANS is possible to look at how the arrangement of the biopolymers, such as cellulose, lignin, and hemicellulose in the plant cell wall. So where the uniqueness of neutron scattering comes in, so I think I hit, okay, so where the uniqueness of neutron scattering comes in, it's the way it interacts with different elements. So with X-rays, the X-rays scatter off the electron cloud, so they're actually sensitive to the larger Z elements, whereas neutrons scatter off the nucleus, and you can observe the lighter elements with neutrons. So and this is especially the case with hydrogen, and then they also interact with different isotopes in different ways. So, for instance, hydrogen and deuterium have very different scattering cross sections, and that allows us to do some very unique types of experiments where we can vary the contrast within a particular system. So, for instance, if we had a protein in a lipid assembly or a detergent micelle, we can actually look at the detergent or the lipid protein complex, or we can look at the protein within that lipid complex by essentially masking the scattering contribution of the lipid. That's achieved by varying the ratio of H2O to deuterium oxide in the solvent system. Similarly, we can vary the contrast of a protein DNA or RNA complex to see the individual protein and RNA and how their conformations change in a complex. If we actually biochemically introduce deuterium into the backbone of the protein, that then allows us to look at how proteins interact with each other in complexes. So it's a very versatile technique, and it's most impactful when we think about using it in combination with other structural techniques, such as NMR, cryo-EM, X-ray scattering, and diffraction. So because the ability to distinguish different biomolecules by labeling is so central to a neutron scattering technique, CSMB also operates a deuterium labeling facility. So we offer a variety of capabilities to support the neutron scattering program, and these are all available to users who come to use our facilities. We can do large-scale fermentation of yeast and E. coli. We produce both hydrogen and deuterium labeled proteins in both eukaryotic and prokaryotic systems for both soluble and membrane proteins. More recently, we have added the capability to do chemical deuteration, so we now can offer the labeling for lipids, detergents, and other small ligands. And of course, to support the production of these proteins, we have facilities for purification and characterization of deuterated proteins, such as light scattering and Sachs. So these are two Neutron facilities that are at Oakhurst National Lab. We have a high flux isotope reactor, which is a reactor-based neutron source, as well as the spallation neutron source, which is an accelerator based neutron source. The Biosands instrument is operated from the high flux isotope reactor. So just in summary, SANS can provide some unique structural insights into a wide variety of biological systems through targeted isotope contrast. So I showed where you can actually go all the way from looking at individual biomacromolecular complexes to looking at very complex systems such as soils or lignocellulose. It's best used in combination with other structural tools. You can find out more information about the structural tools that are available through Be Your Structural Biology resources at the Be Your Structural Biology portal. The link is here. You can find out more information about the specific capabilities the CSMB offer at this website, or feel free to contact me directly. So thank you very much for your time. And then I'll hand you over to Mike St. Clement now from NEON.
Great, thank you. So I'm gonna switch gears a little bit, everyone. Hopefully you can hear me clearly, and give a really quick high-level overview of NEON's field sites and data products, which is more aimed at giving you the resources to sort of dive into what's available from NEON as far as the archive samples and the biorepository go, and the data products that are associated with them, and be able to start digging in and then have the information you need to ask questions further of us as you develop a proposal. So just if you're not familiar with NEON's NSF-funded project operated by Battelle, we have 81 field sites distributed across the continent from Alaska to Puerto Rico. That's 47 terrestrial sites and 34 aquatic sites. And throughout all these sites, we have a series of instrumentation as well as sampling, which occurs, feeding over 180 data products. We're just over that now. We just added methane concentrations at all of our flex towers are now available throughout the entire network recently. So that brought us up a little bit higher. I think we're at 182 now, actually. And along with that, we also collect a lot of archivable samples, collecting over 100,000 samples per year, which are fed into the NEON biorepository. And so NEON overall is comprised of sort of 3 distinct data collection methods. You can think of it as there's automated instrumentation, and that's like our flux towers, as well as all the meteorological instrumentation and soil instrumentation surrounding those towers. There's observational sampling, and that's what we're going to focus on today, because this is what we think of and we define observational sampling within the NEON program as samples that are collected by people and then processed in a laboratory or sent to our archives. And like I mentioned a moment ago, the biorepository is the main warehouse for over 100,000 samples that NEON puts in archive each year. And so we're going to talk really quickly about finding what you need there and how you might get these different soil samples. And just for your information, we also have an airborne remote sensing platform, 2 Twin Otter airplanes, which fly each of these sites on a yearly basis during peak greenness. And you can learn more about all of these on our website. So like I said, in each NEON site, this is a really basic caricature of NEON site. For the sake of time, it will kind of help you understand. We have a flux tower, which we kind of think of at NEON as anchoring the research site, the terrestrial research sites. The darker green color here would be the flux footprint. And then we have a bunch of what we call distributed plots and tower plots. The tower plots and distributed plots are fairly similar. There's some differences in what's collected in them, but basically the tower plots are plots which exist within the tower footprint. And the distributed plots are a similar set of plots, which are distributed on quite large areas. So some of the NEON sites are up, those plots are distributed over several kilometers. So you can get quite a bit of heterogeneity, quite a bit of samples. But what I want to get across here is that each year, we collect samples from a number of these plots, 10 plots, and we collect 3 surface samples per plot. And each one of those samples is, those 3 surface samples are homogenized. They're picked clean of coarse roots, rocks, and debris, and they're transferred into 5 by 5 milliliter cryogenic tissue vials. And that soil is frozen at -80 and sent into the bioarchive, where it resides and is freely available for anyone interested in working with soil to request and use in conjunction with their own research, or specifically in conjunction with this FICUS call. And so that provides a really good foundation for conducting a lot of this research, where you don't need to be going out there to collect the soils yourselves. And so each one of those vials contains about 0.4 to 0.6 grams of organic soil, or 1.5 to 3.5 milligrams of mineral soil. And that's 3 samples per plot for the 10 plots. And so we have quite a bit available. And so one of the things here, is figuring out how to sort of consider what you might want to use in this research. So I'm going to give some really high level steps for planning, and then you'll have the information to reach out further to Neon by the end of us in contact once you get sort of a framework for what you're thinking. So the first step for planning is really to visit neonscience.org if you're not familiar with it, and just look at the map of our field sites. Like I said, we have 47 terrestrial sites distributed from Alaska through to Puerto Rico. And so you can go through and look at these field sites, look at them either a list or a map, and start thinking about the ecosystems with which you might be interested, the forest types you might be interested in, the climate, et cetera. And so once you have maybe a rough framework of a number of sites you might be interested in working with, and to see if you want to get samples from there, what you can do next is you'll be able to go right over to the biorepository. You can actually click right to it from the top of the Neon webpage. And you'll come over to the biorepository webpage for Neon, and you'll see right in the big banner at the top front of the biorepository webpage will be a sort of a search bar. And you can just type in the word samples, and what will come up is you'll get a huge sort of grade spreadsheet here like this. I think right now there's over 103,000 samples of soil in there, records of soil. And so you can get those, you can filter them. And if you click on any individual record in there, you can start to filter by the site code, or you can click into it and you'll get a record of like the location, what site this was collected at, where, when. You can click into a map and see where that came from. And so you can start to sort of filter and build a list of samples you might want to request from the bioarchive or biorepository for this work by digging into the website that way. It's really quite useful, and we'll get. And so then from there, you might come up with a list of samples you want to work with. Say you want to work with samples from Alaska and samples from Harvard Forest in Massachusetts. So perhaps you've got a list of archive samples, and you want to see what other data might be collected there that's related to soil microbes or soil in general. You can go also to our data.neonscience.org, which is our data portal, and start filtering by the word soil, for instance, and you'll see all the data products that are related to soil that are generated by Neon, and then you can search by sites and pull all the ancillary data that's related to those samples you have, and sort of see that grand map of what's available. So what can you get from Neon versus what you can pull from the biorepository, and how you want to extend that research. So it's a really good way to start diving in. Finally, one thing that we do require is that we require you to get a letter of support for the biorepository and to be in communication with them so that they're not surprised by any requests. So you'll want a letter from them. It's fairly easy to do. And so at neonscience.org, at the top, there is a resources. Not sure what's happening there. Oh, there's a resources tab, and then a research letters and assignable assets. And you can follow that link until you come down to this requesting the correct type of letter. And you can see here that there is a type of letter for the biorepository, and you can click on that. That'll put you right in touch with who you need to get in touch with at Neon and the biorepository to submit your form to request a letter of support along with any samples you'd want, and have that done in a timely fashion for submission of these proposals. We can work with you pretty quickly. And additionally, there is always plenty of places on the Neon website and the biorepository website to click through and put in a request for help, and that will all filter down to the correct scientists, and we will respond to you. But really today, just a couple 4 minutes I wanted to make sure that you had a quick sort of sense of how you go through this, looking at the sites, visiting the biorepository, visiting the data portal to see what else is available, and then visiting this sort of assignable assets research support program to begin that process of requesting a letter for this. And anything else that you need above and beyond that, we are happy to help. Thank you so much.
Right. It's my pleasure to tell you about some exciting new capabilities that we're offering through the FICUS proposals. I'm Trent Northern. I'm the Metabolomics Science Program Lead at the JGI, and then I'm going to be sharing this talk with Kirsten Hoff-Markel, who's the Microbiome Science Team Lead at EMSL. So as part of your FICUS proposals, you can now request fabricated ecosystems. So kind of building on what Michael was just talking about, we have these amazing resources that you can access for different field sites around the world. And there's also a great number of resources in terms of model organisms. But what's been missing is something in between, where you can do mechanistic studies of plants and microbes and soil systems within the laboratory to try to bridge the gap between model organisms and field sites. And this is something that we and a number of other scientists have been developing over the last 5 years or so. And we've hosted a number of workshops on this topic, as well as various sessions at conferences. And there seems to be broad support for the need for model microbiomes. And out of one of these workshops, which we call the Ecofab Summit, we developed this vision of how fabricated ecosystems could really enhance microbiome science by providing standardized systems that scientists around the world could study, could reproduce each other's results, and could collaborate to apply various modalities to study these systems. And so part of it is creating these reproducible habitats, making collections of plants and microbes, et cetera, that scientists can access, creating all the standardized protocols. And for the FICUS protocol, FICUS proposals, this is all standardized because you're getting this done as part of your FICUS proposal. Also having the capabilities to look at the spatial temporal dynamics of these ecosystems. And once these are established, then you can investigate mechanistic studies and use machine learning across studies. And in line of consistent with this paper that was defined by the workshop, we formed a steering committee that oversees a number of different activities, including intercomparison studies to establish the reproducibility of these systems. And we've also developed this device, which is the one that you can access through the FICUS proposal, which is what we call the plant microbe Ecofab. You can see a picture of it here. It's about the size of it has the base of a 96 well plate, so it's about that big. And it allows the sterile growth of plants and inoculation and characterization of microbes growing within this chamber, both hydroponically and also within soil systems. You can do a number of things with this, including performing metabolomics analysis, doing sequencing. You can even do gas analysis for volatiles or isotopic tracers. Here's some examples from the Mortimer lab doing really beautiful biogeography studies to look at the localization of bacteria along the root surface. This Ecofab is really designed around Brachypodium distachyon, which is the JGI flagship model grass. It's relevant for almost all bioenergy crops as well as grain crops. JGI has a number of resources for this, including thousands and thousands of sequenced mutants. People have grown all kinds of different plants in these Ecofabs, from camelina all the way to mangroves. And here's a picture showing that you can grow plants and study soil systems as well. We did perform a ring trial intercomparison study where Joelle prepared kits that she sent out to 3 other labs. So we had 4 labs total with Ecofabs, seeds, different media, and all of the labs performed the same experiment to test the reproducibility. And what we found is that across each of the labs, we saw very consistent results. So here you can see these bars, each are from a different laboratory. So we had a reproducible significant changes in fresh weight in the roots and shoots in response to the different media that the plants were grown on. The metabolomics results showed consistent tissue metabolites where they cluster by treatment and not by laboratory. And the same thing for the exudates. Being able to actually image the roots is a very important component of the Ecofab device. And we saw that consistently across all of the laboratories that growth in a soil extract promoted the length of primary root hairs, as you can see here. And also you can use these systems to study plant microbe interactions. And this is a simple example using switchgrass, where if you grow the plant by itself, you can see what it's releasing into the environment. And then when you start adding different microbes with the plants, you can, so each of these are different metabolites here. So this is what's being produced by the plant when it's grown by itself over time. As you add in different microbes, the microbes consume many of these metabolites, but they also start producing metabolites. So you can start looking at these ecological interactions here. And we're really excited to see, for example, serotonin production, which when you add serotonin to switchgrass, you actually get longer roots. So that's the plant microbe Ecofab. And now I'm going to hand it over to Kirsten to tell you about the soil Ecofab.
Thanks, Trent. I'm Kirsten Hofmackel, and I am a scientist at PNNL, and I work on soil microbiome science. And we've been collaborating together on these Ecofabs, and the soil Ecofab is designed in order to have a structured porous media. That's good for studying soil microbial interactions. And as Trent was discussing previously, these are reproducible, they're scalable across different laboratories. So we can start to generate a community of scientists that are developing deeper understanding of how microorganisms interact with each other and with plants to confer some of their many important functions. And so the goal of the soil Ecofab is to be representative of a soil habitat, and that it has some structure to it, unlike some of the hydroponic systems, that it's flexible for diverse measurements, and that it's compatible with the strengths of the user facilities. So this brings us to sort of the grand challenge in soil microbial ecology, which is dealing with the heterogeneity of the environment. And so a soil core has lots of microbial community hotspots. And so we're trying to define these reduced complexity systems where we can test hypotheses and try to move beyond some of the correlative understanding that we can generate from working in soil, and try to identify specific mechanisms that we may be able to demonstrate in these reduced complexity reproducible systems. And so similar to the way we've learned so much from E. coli, we can work in these systems with model organisms or model communities in a standardized habitat in order to rapidly advance our understanding of how microorganisms interact with each other and the environment. So in order to do this, we've developed these soil Ecofabs where we have a porous media there. So we have two different structures. One is like a rough aggregate structure, and the other is a fine porosity, in order to look at how spatial structuring can influence microbial functions. We can manipulate moisture in these. So many of our microbial assays are done in liquid culture, which we know the phenotypes that are expressed under reduced moisture conditions can be quite different. And then we've functionalized these devices so that they're compatible with many of the imaging capabilities at the national user facilities. So if you use these devices, you can do bulk measurements like biomass or fluorogenic assays that are compatible with things that are typically done in soil assays. They're also completely compatible with any of the omics analysis. So you could do proteomic, metabolomic, or lipidomic analysis, as well as transcriptomic analysis. So you could extract your DNA and RNA for sequencing. We can also do spatial analysis through microscopy. So you can look at the phenotypes or interactions of different organisms, as well as MALDI mass spec imaging, which will give us spatial resolution of metabolic interactions. So basically, you could take your microbial community, we can inoculate them into different ports, for example. Then you can add an experimental treatment, be it the substrates that you add or the moisture content, temperature, and then we can evaluate these different phenotypes and do profiling using omics analysis, as well as generate data that could then go into NMDC for some of the modeling efforts that are ongoing at KBase and in other lab efforts. So these are just some examples of experiments that we've done with some fluorescently tagged model soil consortium that we've developed. So these are organisms that are native to our field site. We've isolated them and put protein tags in them so that they fluoresce. And here you can see how we can look at different phenotypes under different conditions. And the circles there are these pores. And so we can inoculate different organisms at different ports and see which organisms interact with each other in response to different environmental conditions. Here you can see that with the different porosities, we see quite different phenotypes in terms of the way that microbes interact. And then we can subsequently take these for example, MALDI imaging to look at the metabolic interactions to complement these differences in spatial interactions. And then finally, we can put all of these together for this FICUS call, where we have complementary resources at EMSL and JGI, and Trent and I would both be happy to answer any questions that you have regarding this.
Thank you, Kirsten and Trent. I'd like to finish just by going through a couple of tips and tricks that we found through the years that help to submit a competitive proposal. So the first one is in preparation to come into the process prepared. The first thing we found that really works is generate a compelling hypothesis and to clearly define your science questions. Proposals that don't typically work are the ones that come in with a laundry list of analytics that you want to do on a particular system. Reviewers like to see that science question. They like to see the clear, definable, actionable hypotheses to frame your idea and say, this is what's novel, this is what's new, get them excited about it. What are you trying to answer? Why is the approach you're proposing the best way to get there, or a way to get there? And then why do you need these facilities? How will your research benefit by having access to these facilities? Contact the facility staff. If you're using JGI, EMSL, NEON, Biosands, whatever facilities you're using, you can contact us before submitting your LOI. We are here to help. We're here to help scope your questions so that when you're submitting even just the letter of intent, we can look at it for what is the feasibility? What is the scope? Can we actually do your experiments? But even more, we can also say, well, have you thought about doing these other things that could augment your experiments, things you might not have thought about? And all of our experimental staff are very knowledgeable in the type of analytics that they represent, and so can really help you hone that science questions so that you really are being able to accelerate your own science. And then I can't emphasize this enough to please start early. If you reach out to the staff, the earlier you reach out, the more attention you're going to get. As we get closer to the deadline, we get more and more requests for help, which means we can spend less and less time for every person. So if you want that feedback, reach out early and keep an eye on the deadlines. Make sure that you know not just the days of the deadlines, but the times of the deadlines. We have one call that had closed at midnight or 11 o'clock, and now it's closing at 5. So keep an eye, just not on the dates, but also the times. So some elements for a strong proposal are, and I mentioned this before, have clear either scientific questions or hypotheses. You don't always have to have an actionable hypothesis, but you do have to have a question. You can't just say, hey, I work on this plant. I'd like to do all of these analytics. Why are you doing this? Be clear in your experimental design. What samples are you going to be sending? What facility resources do you want to access? What are you going to measure? And how are your measurements going to link back to that scientific question? If you're using multiple sets of instruments and facilities, be clear on the data you're going to be collecting for each of the samples. And then it helps us to have a clear timeline for sample collection so that when we're trying to plan these, we know what's going to come in when. And this is a big one. Provide a measured assessment of the potential benefits of your research. So don't stop at, I'm going to get all this data. Add something in there, and this is how this data is going to be used in this model, or here's how it's going to answer my question. Here's how it's going to accelerate my research. We have a requirement that has that the research is relevant to BER's mission, so you can point back to the call as well as BER's mission within your proposal, and just familiarize yourself with BER's mission. I mean, it's environmental research, so if you're going to propose something on tumor samples, you're probably not going to get through the review process. And this is just a global tip, is use understandable language. When you write a proposal, assume the person doesn't understand every aspect of what you're proposing. Take it up a level, make sure that the reviewer that's seeing it that may not even work in your area understands what you can do. Focus on comprehensibility, don't use jargon, don't use a whole lot of acronyms. If you have a proposal full of acronyms, you get confusing and people have to go all over the place to figure out what you're trying to do. Look at educating your reviewer. Now, these aren't very long proposals, but educate as much as possible to bring them along with you and to bring them along in your discussion. And make sure your proposal is well written. One of the biggest mistakes I see is that people put a proposal into a BER call or an NSF call, and they say, "Oh, I'm just going to take the same language, just cut it so that it fits into the page limit, and then submit it." And then when you actually go back and read the proposal, you end up missing important elements, or it doesn't hold together because the place you've cut out explains an important part of your research, but it didn't fit. So don't just condense, but rewrite and reformat so that it is a good, compelling, understandable proposal for the reviewers. And I think with that, we are willing to take any questions or that you might have. And we thank you all for your attendance and attention.
All right. Well, thank you everyone for your wonderful presentations. And so now we're going to start the Q&A portion of this webinar. We did get a couple questions that came in. And so again, if any of the folks in the audience have questions, please drop them in the chat and we will grab them and let the panelists know. So, okay, let's talk about one of the questions that we saw come in. What are the data policies of the different facilities? Can Tanja, Mary, Hugh, Michael, can one of you tell us about your, or we're gonna have to go through the rotation. Can you tell us about the data policies?
Yeah, I can get started with JGI's data policy. So at the JGI, for sequencing projects and for metabolomics projects, the data are subject to a 1-year embargo. Now, what does this mean? That means that the data is only accessible to the PI and any co-PIs or collaborators for this first year. And just to be clear, that the clock starts to tick when the project is absolutely complete. When we completed the sequencing, the analysis or metabolomics analysis. It's kind of the whole package. And then the 1-year embargo starts. For DNA synthesis projects, this embargo period is 2 years. And that's our JGI policy. And then after that, after the embargo expires, we will make the data publicly available on JGI's portals, as well as push the data to NCBI. So it's accessible for NCBI as well.
So just, I mean, ours, the EMSL data policy is very close to the JGI because we've tried to align them. But our data policy is that it's 1 year from creation of the data, not from closure of the project. So as soon as we generate the data, there is 1-year embargo, and then it is available to be released.
I can go next and speak on behalf of NEON. For ours, we typically provide the PIs the guidance that whichever agency their project and work is funded through is the agency that those guidelines will be followed. So for instance, if you were to receive NSF funding, you would follow their guidelines. If you have data from EMSL, it would follow theirs. We do ask, however, that if you were to use any of our data products, which are openly and freely available, you cite them, and I can put in the chat here a link to our citation policies for the other data that's already available, and that would include, I'll put that in here for everyone.
And we would be similar to NEON. We would ask the users to follow the funding agency's requirements. We don't have an embargo when we would release the users' data, that they keep control of their data in that sense.
All right. Okay, so the next question, let's see. I saw pictures of Arabidopsis roots shown during Trent's section. I just wanted to verify that using Arabidopsis as a model organism for larger environmental questions like to study root microbiome interactions, is within the framework of BER's mission?
That's a great question, and you have a very sharp eye. Good job. I'll just take a quick stab at this, and then also let Tanja jump in if I've missed anything. So if you're asking a very fundamental question that relates to DOE BER questions, and Arabidopsis is the best system to address that question, then that is within scope. Tanja, anything you'd like to add to that?
I need to unmute myself. No, you answered it perfectly, Trent. Yeah, we would consider that within the DOE mission, within the BER mission.
And just to add, we've actually funded projects on Arabidopsis and Brachypodium as model systems, as long as your broader question is something that is BER relevant.
And we've grown a number of model plants in these systems, including Cateria and others. And you're very welcome to reach out to me if you have questions about the suitability of, whatever system you're working on to use these, for these kind of devices.
All right. So for Michael, if I want to speak to someone at the biorepository, who do I contact? And how do I find that information?
Yeah, sure. That's actually a great. The easiest way to do it is just go to neonscience.org and click the biorepository tab at the top, and that will take you over to their webpage, which will have multiple places where you can go ahead and connect. The main contact for the biorepository, though, is Kelsey Ewell, and we can go ahead and get her email provided in the chat here in a minute. But I can send that. But you can also, another really easy way to access that would be to just channel through Neon. If you go to the Neon Science homepage and click resources, you'll see a part for research support, and there's a bunch of contacts there, and reaching out to anyone through Neon that way, we can also connect you. So there's multiple ways to get there, but we can put that in the chat. Thank you.
Excellent. Yeah. Let's see. So do you have to use all of the facilities when you submit a proposal, or can it just be some?
So I guess I'll take that one. You don't have to use all the facilities. So you don't need to envision a project that uses Neon and Biosands and JGI and EMSL, but you do need to at least use 2 of them. And my advice is not to say, I'm going to do all my sequencing in JGI, and oh, by the way, we want to get, we want to do something at EMSL, or we want to do something at Neon. They have to be equally weighted, or it doesn't come across as a balanced proposal, and it's harder to get through the letter of intent stage. So you have to use at least 2 of them and make the facilities as balanced as you can.
All right. When is a good time to reach out to facility staff?
As soon as the call comes out. Anytime before the deadline, but as soon as the call comes out, and as soon as you have a nucleus of your idea. You don't even have to have a fully fledged idea. You just have to say, even if you had come to us and say, I'd like to do this, we can help even nucleate a scientific question based on some of the analytics we have. And which, again, the sooner you reach out, the more attention you're going to be able to get from the facility staff.
Okay. How do I request Ecofabs as part of my FICUS proposal?
I would recommend that you reach out to me for the plant microbe Ecofab, and then Kirsten for the soil microbe Ecofab in advance of submitting your proposal. You can just include it directly in your FICUS proposal, but I think it would be best, if possible, to reach out to us and just kind of check in on the application, etc. Kirsten, anything you want to add to that?
No, I think that's great. I guess I would say that, that can be one, as Mary was saying, that could just be one component of your proposal, right? So you might be doing some pot studies with your plant in the soil, and then you may do some Ecofab experiments and complement it. So I think it could be one component. It doesn't have to be the whole proposal.
Yeah, great point.
All right. So Michael, back to you. In addition to the biorepository, are there other soil archives available?
Yeah, that's a great question. Actually, yeah, there's quite a few. So the biorepository does have and contains all of our -80 samples. But we also have a number of other soil samples. We have horizons, samples from every horizon in the NEON megapits across all 47 terrestrial sites that are air dried and stored at NEON headquarters. And there's also the potential via NEON, if you would like additional sample that's not currently available or in the archive or in the biorepository, that you can go through our assignable assets program to have our field technicians grab a little bit of sample when they're out there doing our sample and split that and pass it along. So I can put another link in the chat, which is the NEON sort of assignable assets research user support program. And if you have any questions that are beyond the biorepository or any just bizarre questions that you're not sure really where they fit, accessing us through that page is the best, and it will filter to the appropriate people. We've kind of set up a lot of redundancy, so it will always get to where it needs to be. But that one's really a good catch-all, so I'll go ahead and throw that link in there. But yeah, there's quite a bit of other soil available.
All right. Let's see. Has FICUS previously supported deep subsurface research, or is it focused on just surface environments?
I can take that one. So I don't know if we've had a lot of subsurface specifically through the FICUS, but I know JGI has done a lot of subsurface sequencing of, for example, subsurface aquifers, etc. It is well within the DOE mission. So it could be, if it's pitched in a DOE relevant way, it would be very well within the proposal call. And I also want to point out that we do have on the website, we do list all the successful FICUS proposals from the past many, many years. So if you're interested just to kind of see what we funded in the past, it's worthwhile just spending a few minutes browsing the website and seeing the types of proposals that have been accepted. But yes, I would say these are really DOE relevant systems.
Right. And Hugh, one for you. What type of data analysis capabilities are offered through the Center for Structural Molecular Biology?
So we realize that small angle neutron scattering isn't a typical technique that people use. So we offer support for all aspects of the experiment. For sample prep, I mentioned deuterium labeling of biomolecules for your experiment. And once the data is collected, then we have a variety of analysis approaches, depending on the type of system you're using or studying to analyze your data. So we have analysis tools for biomolecular complexes. We have other analysis tools available for the more complex systems like soils and lignocellulose. So we can help with all aspects of the proposal. And if you contact us before you submit it, we can work that into the proposal as we write it.
All right. And we have time for one more question. So what type of analysis capabilities are offered at JGI?
Yeah, so at the JGI, we won't just give you the raw data. We will process that data quite extensively. We have a lot of different analytical pipelines. We do extensive QC. We use state-of-the-art tools for assembly and, for example, for data mapping, depending on what you're asking for example, for transcriptomics, for counting applications, we will map the data to a reference genome. We also have what we call our kingdom portals, which is IMG, the Integrated Microbial Genomes platform, as well as microcosm, picocosm, phytosome, for all the different kind of domains, so to say, where we integrate our plant genomes, fungal, algal genomes, bacterial, archaeal genomes, metagenomes. They're being annotated and then integrated with other genomic data sets for comparative genomic analyses. And then, as I mentioned a little bit in my presentation, we have pipelines, for example, for resequencing, for evolved laboratory populations, for example, where we would provide you with a table of SNPs and small insertion deletions. So we do provide quite some analysis tools.
Great.
And for metabolomics, actually, Trent can comment. I'm going to let him quickly chime in.
Well, just very briefly, yeah, we do. We provide both targeted and untargeted metabolite analysis. The targeted analysis is based on authentic standards that we've run on the same instrument. And we have about over 5,000 standards that we use for annotating metabolites. And then we also make the untargeted data available in a number of ways, but especially using GMPS, which is a very common metabolomic service. And this allows you to discover new molecules. And we also have some tools for integrating it with other types of omics data.
Great. All right, well, with that, we are at 1 o'clock. And so I want to thank everyone for joining us. Thank you for all the presenters. We hope that you have, this has clarified any questions you may have had about this open call for the EMSL JGI CSMB and NEON FICUS call. Again, if you have any other questions, please feel free to reach out to any of us on this webinar. And next webinar is on February 23rd on subsurface science capabilities at EMSL. So please make sure that you're able to join us. And you'll also be receiving an email that will ask your, oh my gosh, I'm getting all tongue-tied right now. It's to ask your feedback on how this webinar went for you. So please, if you could fill that out, that would be great. It helps us to get better each time. Have a great day, everyone.